So there have been different developments in the molecular biology, so that paved the way for sequencing. So we got special polymerase enzymes. There was the discovery of the gel electrophoresis, as well as for the amplification of the DNA, the PCR technique got invented. So all of them, they helped in the finding the better approaches to do this sequencing. So here, for instance, we just have a review for DNA synthesis or replication in order to elaborate the action or the role of this DNA polymerase. So on the back end side, you see a template. So template is actually the strand which is actually red, and it's complementary to this, a new strand is prepared. So actually this bottom strand is what we are actually having over here. So the template is red, and the new strand is formed as a complementary to this. So this enzyme DNA polymerase we see is actually synthesizing the new strand, which is blue color here. And then if we look into the directionality, the template is actually arranged in 3 to 5 prime direction, whereas the new strand is synthesized in a 5 to 3 prime direction. So new nucleotides are actually added on the 3 prime end of this new strand. So polymerase is the enzyme that actually incorporates or actually polymerizes those nucleotides together while getting the order from the template strand. So in DNA sequencing, we need a special polymerase, so that has high processivity. That means that it should not go away while under normal circumstances, the polymerase sometimes it gets off from this replication process, and we actually want some longer fragments, so we want high processivity. Secondly, normal polymerases, they have 5 prime to 3 prime and exonuclease activity, which is there actually to play some important role with DNA repair. But during this sequencing, we don't want this activity, so we actually need a special polymerase that is called as a clinopolymerase. So if we take away that 5 prime to 3 prime exonuclease activity, the remaining part we call it as a clinopolymerase. So that is used in this sequencing. Basically this clino is actually derived or obtained from E. coli. It is modified DNA polymerase 1, and it lacks that 5 prime to 3 prime exonuclease activity. But the issue is that it has a relatively low processivity. So there is an improved one, that is sequinase. So that has been derived from bacteriophage T7, and that has high processivity, and it has a rapid reaction rate, and it has a negligible exonuclease activity. So we can use modified nucleotides also as far as as a substrate during this sequencing process. So let's see how these nucleotides are added into a growing chain of DNA during the process of replication. We see in this diagram, this is actually the terminal or the last nucleotide, which is part of this growing chain. So we see different carbons of this, in this 5-ring carbon sugar is there in the middle, with which on position number 1 the base attaches right here. Position number 2 is where it is deoxygenated, so that's why we call it as a deoxyribose nucleic acid, because of in the nucleotides the position number 2 is deoxygenated. Position number 3 is critical, where the incoming nucleotide attaches itself, and position number 5, which is this corner thing, is actually position number 4 right here, and this is actually the carbon number 5 with which it is been attached to the previous nucleotide. So the incoming nucleotide, which is actually a trinucleotide, dideoxy trinucleotide. Here we see the nucleotide that is coming to attach with this nucleotide chain. It's actually a triphosphate. Basically we label it as DNTP. It is a deoxynucleotide triphosphate because there are 3 phosphate groups which are present. So 2 of them, they break away right here, and then they are relieved as a pyrophosphate molecules, and then this nucleotide attaches to the 3 prime position of the previous nucleotide. So presence of this OH group is critical or is important for the attachment of this new nucleotide to the growing chain. So here we see a special nucleotide, so that is actually called as dideoxyribose nucleotide. The difference is on position number 3 there is no OH group, it's just an H, so it is deoxygenated at two position number 2 and position number 3. So that's why it's called as dideoxyribose nucleotide. So when this kind of nucleotide gets into the incorporated, there is no room for the new nucleotide to come and attach there. So they kind of terminate this process of replication. So that's why we call them as terminators. So discovery of terminators is also important in sequencing. Here is just an example. So that is the termination using those terminators. So we put the mixture of two types of nucleotides. So we put the normal guanosine triphosphates and we put the dideoxyguanosine triphosphates. So this sequence might generate mixture of different chains which are terminated at G position. So we can have, for example, 1 to 6 different possible fragments. Gel electrophoresis is another important technique that has contributed a lot in genome sequencing. What we do here is we take those different size fragments of the DNA and we separate them on an agarose gel. So here is for instance the shape of the gel which we see, and we let this DNA, we then provide the electric current to the mixture, and the DNA is then it runs, since it's negatively charged particle, so it runs from the negative to the positive charge. So while it's running on the gel, different fragments they are separated based upon their running speed. So obviously that depends upon their sizes. So they run from this negative to the positive terminals, and then the gels are right from here. So here are the smallest particles which are running faster, and then bigger, and the biggest ones, they are in the end. Samples are loaded here in the wells, and then while looking into the comparative sizes and while looking into the what position they are, we can determine the sequence of the nucleotides from here. So what we have seen so far, that there are some significant advancements in molecular biology, so that has been helping in improvements in this genome sequencing technologies.